Hello everyone, my name is Fernanda Rosa and today I will share with you some key gene networks and pathways identified in lactating dairy cows with soil ulcers. Lameness is a major economic loss on dairy farms, and soil ulcer is one of the three most common causes of lameness affecting dairy cows. Soil ulcers is a disease that can damage the corium and affect the normal keratinization process of the hoof tissue, as well as soil ulcers can prone cows to develop other conditions, including chronic lameness and systemic inflammation. And the most common soil ulcerations are found on the hind lateral claw, as shown in this photo. And when we talk about systemic inflammation, it's because a local acute inflammatory response, such as soil ulcer, can release pro-inflammatory cytokines into the bloodstream, including interleukin-1, TNF-alpha, and interleukin-6. And those cytokines, they can signal the liver that there is an inflammation. And the liver will respond to that stimulation by increasing the production of the positive acute phase proteins, such as serum amyloid A, hepatoglobin, and ceruloplasmin, while the hepatocytes will decrease the production of negative acute phase proteins, such as albumin. Therefore, the aim of this study was to evaluate the transcriptomic profile of the corium tissue of lactating dairy cows experiencing soil ulcers disease. And to address that, we use multiples hosting dairy cows select for the experiment based on their condition of healthy cows that had no other underlying metabolic or inflammatory conditions, or cows identified with soil ulcers. We assessed the lameness based on locomotion score from 1 to 5, where 1 is equal to normal and flat back, such as this photo, and locomotion score of 5 is extremely lame cows, such as this other photo here. We also measured the hoof hardness of those cows using a durometer, as shown on the left picture, and we use the average of 3 different measurements on the sole of each cow as the hoof hardness. We also used infrared thermal imaging system to measure the surface temperature of the hoof from each cow. And the hoof biopsy procedure was performed as previously described by Osorio and collaborators in 2012, which briefly, the cows were restrained in a hoof trimming chute, the limb was clean and anesthetized, and we use a sterile drill bit to drill a hole through the hard cornified tissue in each sole, and we use a biopsy punch to cut through the remaining hoof wall and the underlying corium, and a sterile scalpel blade was used to finish removing the biopsy sample, and all the samples were frozen in liquid nitrogen. For the RNA isolation and sequencing, we use commercial kits. The RNA isolation was also performed using a bit bitter homogenization, and all the RNA samples were sequenced using Illumina instrument, and using our software, we analyzed the differentially expressed genes, and we also perform gene enrichment analysis using gene ontology terms. Prior to each hoof biopsy procedure, we also collect a blood sample from all the cows to measure blood biomarkers of liver function, inflammation, and oxidative stress. Here are our first set of results, where for the group of healthy cows, the locomotion score average was equal to 1, while the average locomotion score for the group of cows with sole ulcers was 2.5, and we observe a higher hoof hardness for the group of healthy cows compared to the cows with sole ulcers. The infrared thermal imaging analysis demonstrated that there is an increase in the temperature surface of the sole in the cows with sole ulcers compared to the healthy cows. As you can see here on these images on the right, the more yellowish is the image, higher is the temperature, which was observed for the images of the sole ulcer cows compared to the healthy cows. Now, moving to the transcriptomic analysis. Based on the principal components analysis, we observed a robust separation of the transcriptome between healthy cows, the cluster dots here on the right, compared to the cows with sole ulcers, the cluster of dots on the left side of this PCA plot. And this was reflect of more than 10,000 differentially expressed genes in both groups of cows. And to visualize some of these key genes impact by sole ulcers and compared to healthy cows, here is a heat map showing the top impact 25 genes, with the healthy cows on the right and the cows with sole ulcers on the left. And the downregulator genes in the cows with sole ulcers were genes highlighted by these green arrows, which you can see that they were most of the keratinization process genes, such as keratin 34, 86, and so on, while the upregulator genes in the cows with sole ulcers, they are highlighted here by the red arrows, including inflammatory-related genes such as serum amyloid A, as I described previously, some regulatory genes of the innate and adaptive immune system, as well as pro-inflammatory cytokines, which the most impact gene was interleukin 6. And for the gene ontology analysis, first we clustered the downregulator genes by sole ulcers cows relative to the healthy cows, and that cluster downregulator pathways within the cellular component term, including keratin and cytoskeleton filaments, while the gene ontology analysis for the upregulator genes by sole ulcers relative to healthy cows enrich biological process pathways, including extracellular matrix organization and cell surface. Now, associating the transcriptomic findings with blood biomarkers, we observed that cows with sole ulcers, they had greater beta-hydroxybutyrate and non-esterified fatty acids in the blood compared to healthy cows. And we also observed that liver function biomarkers, including total bilirubin and the positive acute phase protein ceruloplasmin, was also greater in the blood of the cows with sole ulcers compared to the healthy cows. And the same pattern was observed for other liver function biomarkers and biomarkers of inflammation, including hepatoglobin, as well as reactive oxygen metabolites and FRAP, that they are biomarkers for oxidative stress. All these biomarkers were greater in the blood of the cows with sole ulcers compared to the healthy cows. So in summary, cows with sole ulcers, they had an increase in interleukin-6, which in our experiment was a cytokine that signaled the liver, and the liver of the cows with sole ulcer compared to the healthy cows increased the production of positive acute phase proteins such as haptoglobin and ceruloplasmin, as well as increase the releasing blood of other biomarkers of oxidative stress and inflammation. And sole ulcers disease in lactating dairy cows downregulated keratinization process related genes, while upregulated inflammatory related genes. In conclusion, our study demonstrated that sole ulcers impact at the molecular and functional level, and we can use these findings for future nutritional interventions to reduce sole ulcer incidence in dairy cows. And with that, I would like to acknowledge Dr. Osorio's laboratory, as well as South Dakota State University and the Altec company for funding this project, as well as Dr. Kamal from the Altec in Brookings, South Dakota, and all the staff from the Hilltop Dairy Farm from Elkton, South Dakota, for helping us to perform the hoof biopsies. And with that, I'll be glad to take any question that you might have in the following Q&A. Thank you.